Hey science enthusiasts, welcome to another exciting episode where we explore the wonders of science through the magic of claymation. Today I'm diving into the fascinating world of polymerase chain reaction, or PCR for short. This technique is a cornerstone in molecular biology and has revolutionized the way we study DNA. But instead of just talking about it, I'm bringing it to life with claymation. This way you can visualize the process and understand it better. Let's get started. Imagine you have a tiny piece of DNA, so small you can't even see it with the naked eye. PCR is like a biological photocopier, making millions of copies of that DNA. This is incredibly useful for various applications. It's super useful in everything from medical diagnostics to forensic science, and even in research labs studying genetic material. First up, we need our ingredients: the DNA sample, primers, nucleotides, and a special enzyme called Taq polymerase. These are the building blocks of our PCR process. Think of Taq as our little molecular hero. This enzyme is crucial for the replication process. Now, into the PCR machine we go. This machine will help us cycle through the different stages of PCR. Step 1, denaturation. This is where we start the process. Picture this: our DNA is like a twisted ladder made of clay. We need to separate the strands. We heat it up to about 95 degrees Celsius and it unwinds into 2 single strands. This is like unzipping a zipper. Next, annealing. This step is crucial for the primers to attach. We cool things down to around 65 degrees Celsius. This allows the primers to find their matching sequences. Our primers, which are short DNA sequences, attach to the specific spots on the single-stranded DNA. They act as starting points for the new DNA strands. If DNA were Velcro, the primers would be the matching hooks and loops. They fit perfectly together. Finally, extension. This is where the magic happens. We heat it back up to 72 degrees Celsius. This is the optimal temperature for Taq polymerase to work. Here's where Taq polymerase comes in. It adds nucleotides to the primers, building a new strand of DNA. It's like our claymation hero is adding pieces to a Lego set, making a complete ladder again. Each piece fits perfectly to form a new strand. And that's one cycle of PCR. But we don't stop there. In reality, we repeat this cycle about 30 times. Each cycle doubles the amount of DNA. By the end, we have millions of copies of our DNA. This exponential growth is what makes PCR so powerful, all from one tiny invisible piece. This incredible technique has revolutionized many fields, from diagnosing diseases to solving crimes and even studying ancient DNA. The applications are endless. It's a cornerstone of modern biology. Scientists rely on PCR for countless experiments and discoveries. And now, thanks to our claymation adventure, you have a visual understanding of how it works. This visual aid makes complex processes easier to grasp. If you enjoyed this claymation explanation of PCR, hit the like button and subscribe for more fun science videos. Your support helps us create more educational content. And remember, science is not just about learning facts. It's about exploring the wonders of the world around us. Keep your curiosity alive. Until next time, keep questioning and stay curious. See you in the next adventure.